Yes, sir. Yes, sir.
Okay, plant physiology. Okay, very good afternoon, my dear friends. So, regarding of this free online coaching, CPGET 2023, JL, DL, and other all competitive exams in plant sciences, we are going to give online mode, free online coaching. Eminent and well experienced resource persons are going to deliver their lectures. So today, our eminent resource person and huge experience botanist, Dr.O.Padmaja Madam Garu, Assistant Professor, Department of Botany, Tara Government Arts and Science College, Sangareddy. Madam, please welcome and start your session. Share your screen.
Thank you, Tirupati sir. It is a good platform for the students, and in summer season, it is very useful for the students.
Madam,
Sir,
any problem?
no, sir, I'm trying. One minute, sir. It is. Sir, is sharing vastaleru? Okay, sir. First of all, I extend my thanks to Tirupati sir for organizing a best platform for the students who are interested in order to achieve the career to getting higher education and the government job oriented courses. Thank you very much, sir. And myself, Dr. Padmaja, Assistant Professor of Botany, Tara Degree College, Sangareddy. Today, we are going to discuss regarding a beautiful topic in physiology, that is phytohormones, and how they are influence of phytohormones in the plant growth, development, and the differentiation of the cells, and how the flowering will be seen, and how all these hormones showing their physiological role in growth and development of the plants, and they are also very helpful in the horticulture and the horticulture and the fruit producing plants. Now, entering into these phytohormones, the phyto means the plants. The hormones are the chemical substance that are produced in the plant itself. So they are called as the phytohormones or the plant growth regulators. They are helpful in the regulating the different types of physiological activities in the plants. So they are considered as a growth regulators also. So growth regulators, phytohormones, plant hormones are nothing but they are the chemical substance they are produced in the plants and alter the growth of a plant or the plant part. So hormones are substances naturally produced by the plants. Sometimes they control normal plant functions such as the root, fruit set, and growth and other development process. So plant growth regulating substance are also called as hormones. They are the chemical substances. In Greek word, they mean the excite. As they are the hormones are producing, they are showing some physiological activities in the plant itself. So before entering into the plant hormones, just if we get a clear idea regarding the plant hormones and the animal hormones, The plant hormones are the simple organic substance. These animal hormones are complex chemical substance, but they are produced by specialized organs called as endocrine. So the endocrine glands are producing these hormones. They are showing the different quick effect in the plant itself. But in the plants, they are not produced by any special organs. They are produced in the plant itself only. In every cell of the plant is having the capacity to produce these growth regulating hormones. And now a brief idea regarding the general plant hormones. They are auxins, gibberellins, cytokines, abscisic acid, ethylene. Along with these, some are recently or at least 10 years or 12 years identified hormones are also there. But we are mainly talking about these hormones. Auxins and gibberellins and cytokines are considered as a growth regulating growth promoting hormones, and abscisic acid and ethylene are considered as a growth inhibiting hormones. And a brief idea, if you see that auxins, the main function of the auxin is the cell elongation. As the cell elongation is taking place, the height of the plant will be increases, and we can see the apical dominance. And due to this elongation, the plasticity of the cell will be increasing. And if you see the gibberellins, these gibberellins are helpful for the cell elongation and at the same time cell division also. That means translate into the growth. Cytokines means the cell, kinase means the division. The cells are dividing, and these cytokines are also helpful for the differentiating the cells in order to develop into a root system also. And it inhibits senescence. Senescence means aging of a plant or the leaves are becoming older. So it also inhibits the senescence. And abscisic acid, the abscission of the leaves and the fruits. That means due to the formation of this abscission layer, the leaves and the fruits are also falling. And if the leaves are falling, the plant is not having the capacity to undergo the photosynthesis, the growth will be stunted. And the fruits, if the fruits are fall, there won't be any yielding also. So it also induces the dormancy of buds and the seeds. If the dormancy is seen in the seeds, there won't be any germination. So it is acting as an anti-role in the plant. And ethylene is considered as a gas hormone, and it promotes the senescence. And epinasty and the fruit ripening. Epinasty means the downward falling and the upward movement of the plant itself. Okay, if you see the some more idea regarding these plant hormones, they are called as the phytohormones, and they are produced in the plant itself. They are chemical substance and organic substance, and they are produced in small quantities. And what is the role of these hormones in the plant is they are showing the growth, development, reproductive process. The reproductive process means the vegetative state is giving rise to a reproductive state. In some plants, we'll see that 3 or be growing. After some time, we'll see the flowering will be seen. So in order to get the flowering also, these hormones play a very important role. And longevity and even the death of the leaves or the plants are also having the role by the hormones. These small molecules are derived from the secondary metabolites. So where these the hormones are produced itself in the plant itself in the biological pathway by the secondary metabolites. And they are helpful for the adaptation of the plants to environmental stimuli also. That means number of environmental conditions we are seeing. For example, stress, salinity of this stress physiology also. These hormones are helpful the plants to sustain the stress and to grow into a plant. A single phytohormone can regulate many cellular and developmental processes, while at the same time multiple hormones often into a single process. That means one hormone is helpful for the single metabolic activities or the development process, or all the a cascade of these hormones roles also play a single process. And there are 6 major phytohormones identified: auxins, gibberellin, cytokines, abscisic acid, ethylene, and along with this we are seeing the steroids also. Most recent they will be include the some jasminates and the strigolactose also. The many research have been done in the steroids and the jasminates and the strigolactose also. Now, if you see this individually by seeing these auxins and their role in the plants, first of all, auxins are the growth hormones, which in a small concentration bring about the cell elongation in the roots and the shoot also. That means the main role is the elongation. As the elongation process is seen, they will be seeing the apical dominance. The term auxin was introduced by Cole and the Hazen Smith. Auxins are synthesized at the growing apex of the stem and the roots. We already see that the apical growth will be seen at the apex of the stem and the apex of the roots. At that place, the more amount of auxins are synthesized. And even that part is not having any microorganisms affecting also. So auxins can diffuse from place to their synthesize to other parts where they promote growth. So they are producing in one place and they are showing their role in other parts also. That means they are diffusing from one place to another place where their role is there. They will moving through this xylem and the phloem also. Chemically, they are some naturally producing auxins are there, and they are artificially production of the auxins are also there. The naturally producing auxins are the indole 3 acetic acid. This is the abbreviation is IAA. And next one, indole 3 acetaldehyde, indole 3 pyruvic acid, IPA, and indole 3 ethanol. So these are naturally producing auxins in the plant itself. And synthetic auxins means which we are artificially producing from the plants. So if there is any commercial use is there, means we will go for the artificial synthesis in order to have the commercial value. For example, indole butyric acid, 2,4-dichlorophenoxy acetic acid is a very important bit, and naphthalene acetic acid. So these are the synthetically or chemically produced auxins. And this is also the 2,4-dichlorophenoxy acetic acid is used as a weedicide in order to kill the weeds. This chemical is very useful. And when isolated auxins from the avena coleoptile tips, that means avena from the oats, they identified the isolated the auxins in the coleoptile tips of the oats. The person is went and he devised avena curvature test. So in order to extract the auxins, he conducted a test. It is considered as a avena curvature test because this test is conducted in the oats. That is the scientific name of avena. It demonstrates the presence of auxins in a liquid condition. The coleoptile tips was removed, the remaining coleoptile stops growth. That means they can conclude that the auxins are producing at the tip of the growing ends. So if the removed tip was placed on an agar block, we already see that this agar block is helpful for the diffusing these hormones into this. Nowadays, these agars are using in order to store the chemicals in the agar block in order to grow the plants in the conditioned temperature also. So if you remove the tip was placed on an agar block for several hours and then the agar block was placed on the cut coleoptile stump, the coleoptiles show typical curvature. So by the diagrammatic, we can clearly understand that these agars are diffusing in this auxins are diffusing into agar, and again they are entering. So the movement will also be seen in this. So as a result, the bent nature will be seen here. If you see here, these auxins in the seedling, that means these auxins are produced at the tip of the shoot, and they are entered into the agar, and they are diffusing into the agar block. Again, these agar block are present, kept on the coleoptile tips. They are showing the bent growth will be seen. That means the auxins are again moving into these coleoptile tips, and they are helpful for the growth of the plant. That means they are showing that they are having the capacity of the growth. So they can conclude that these auxins are the growth promoting hormones, and auxin increase the plasticity of the plant cell wall and involves in the stem elongation. And it was discovered by Wendt. And next, what are the physiological role of auxins? Okay, the auxins are isolated, and what is the role? We concluded that the auxins are produced in the plant itself. They are produced at the growing tips. And what is the role of these auxins in the plants? This is a very, very important point. They may be related for the bits also. So auxin induce elongation of the plant cell, and it is responsible for the callus formation in the tissue culture. That means in tissue culture, due to the totipotency from one cell, we are getting the callus, and the callus is differentiated into root system and the shoot system. That means the organs of forming of organs we are seeing. So this cell elongation, the cell division is very, very important in order to form the organs. And auxins is responsible for the apical dominance. Mainly point is the apical dominance. They are showing that if one plant is having that more amount of auxins are introduced at the tip, their plants are showing the apical dominance. For example, if you see the this Saraka indica plant, they are having eucalyptus. These are showing the tall plants because they are having the apical dominance is more. So apical dominance is a process in which the apical bud dominates in growth and does not allow the growth of lateral buds. That means the suppression of the lateral buds and the dominating the apical bud. As a result, the apical dominance will seen in the plants. And it also influences the differentiation of the xylem and the phloem. That means in the secondary growth, some of the cells have to be formation of this vascular bundles. The differentiation will also be taken place. And auxins initiate root formation. In stem cutting also, if you introduce some auxins to the plants or spare some auxins to the plant, the root formation will also be seen. And we also know that is the suppression of the lateral buds and the apical dominance will be seen here. And auxins promote cambial activity. So this cambial activity play an important role in the secondary growth. So as the cambial activity will be increased, the cambial activity will be increased, and the secondary growth will also be seen here. And here auxins also break seed dormancy. That means the sleeping condition of the seed will also be break by these auxins. If the seeds are in dormant condition, it is very impossible in order to the seeds not be germinate. So these auxins also used to break the seed dormancy. And auxins initiate early flowering and the fruiting. That means formation of the fruits and formation. If we see the flowering only, the flowering leads to the fruits. We'll see that some plants will be more vegetative growth will be there. There won't be any flowers. So this auxins will play a very important role in the formation of the flowers and also the fruiting. Nowadays, every person are growing their plants in their houses for the beautification or for the edible condition, for the eating. So if you are growing a mango plant, but they are not giving any flowers, so what is the use? So for these auxins are internally present in the plant itself, they are helpful for the early flowering and the fruiting. So delay fruit ripening also. They may be helpful in the ripening of the fruit. And indolestic acid induce parthenocarpy. That means the seedless fruits are produced here due to these hormones in tomato, grapes, and the apples. And auxins also inhibits the leaf fall and the fruit fall also. And next one, gibberellins. These gibberellins are also considered as the plant hormones, which stimulate the elongation of the internodes. Whenever this internode elongation will be seen here, the growth will also be seen. That means the stunted growth will be stopped here. So gibberellin was first discovered by the persons like Abuta and Hayashi in 1939. It was named as first is isolated from the fungus known as the Gibberella fusicoria. So he given the name gibberellin. It is a fungus, and from the fungus they extracted these gibberellins. So young leaves of plants, stems, fruits, seeds, and roots of these plants contains these gibberellins. That means whenever these affected by this Gibberella fusicoria, this plant itself is having, they extracted the gibberellin by Abuta and Hayashi. So about 125 gibberellins have been isolated so far. They are named as GAI, GA2, GA3, GA4, etc. So these are having, if you see the chemical structure of these gibberellins, they have a gibberelline skeleton also. And what is the role of these gibberellins? They are helpful for the bolting, means shoot elongation. The shoot elongation will be seen in by the gibberellins in the plants, and elongation of the internodes. These nodes and internodes is a characteristic feature of the stem itself. These, due to this elongation of the internodes, when the aerial parts of genetically dwarf plants were sprayed with gibberellins, they occur elongation. See how they are. That means they are, when the dwarf plant will also, when we induce the gibberellins, the internode will be elongated. As a result, the dwarf plants will be in a gigantic plants will be seen here. That means we can suppress the dwarfism in the plants by inducing these gibberellins. As a result, the internode elongation will be taking place. That means we are also in the bolting. The gibberellins induced bolting means shoot elongation. This bolting will be which we can see. And parthenocarpy, like auxins, gibberellins also induce parthenocarpy in tomato, grapes, and guava also, and breaking of food bud dormancy. So these buds, the dormancy of buds can broken the gibberellin treatment. And synthesis of alpha amylase is a very important one. These gibberellins are break the seed dormancy. As a result, germination will be taking place, and during this germination, synthesis of alpha amylase is promoted by this gibberellin in the alluvial layer of cereal grains during germination. So this amylase enzyme is having the capacity to degrade the seed coat. As a result, the germination or the radicule and plumule will be coming outside, and the seedling will be seen here. And abscission. Abscission means here this abscission layer is formed between the leaves and the stem, or maybe the flower and the stem, or the fruits on the stem. Due to this abscission layer, the early falling of the leaves, or the early falling of the fruits, or early falling of the flowers will be seen here. And flowering in long day plants, it is due to the photoperiodism, means here depending upon the whenever the plant will takes more daylight than the critical daylight, so that plants are called as the long day plants. These long day plants, when they receive more amount of light, the flowering will be seen here. That means whenever the light is fall on these plants for a long time, this will be induces the flowering hormone. As a result, the flowering will be seen in these plants. And substituting cold treatment. By the cold treatment, by the vernalization process also, we can induce the flowering in the plants. That means the gibberellins are also helpful in the flowering in the cold plants also. And the next one is the cytokinins. These cytokinins are plant hormones promoting cell division in plants. And the term cytokinin was proposed by Latham. And cytokines are also known as phytokines. They are the producing in the naturally in the plant, so they are called as the phytokines. And the kinin is the division of the cell. So these they are purine derivatives. These cytokines are the derived from the purines, and they're regarded as the degraded products of ethylene. And root, fruits, endosperm and embryo contain cytokines. So hence cytokines are regarded as the natural plant growth hormones itself. And kinetin and zeatin are common cytokines found in the plants. The main role of these cytokines is the cell division, and it stimulates the division of the cell. That means one cell divides to give rise to the group of cells, and this may be helpful for the organ formation, and is also helpful in the promoting the cell elongation. That means one physiological process is also controlled by the one or two hormones. And breaking seed dormancy. uh, the seed dormancy is the sleeping condition of the seeds. And in order to break the seed dormancy, whenever we treat this with cytokinins, the seeds uh, will be undergoing the germination. So in lettuce and tobacco, this whenever the seeds are going the dormancy, when we treat it with cytokinins, this seed dormancy will be broken and the germination will be taking place. In some plants, years to years the seeds are dormant in condition. So in order to overcome this, when we spare these cytokinins, we can also see the germination process in plants. And delay of senescence. We thought that senescence means the aging, or the leaves are becoming the yellow color. The leaves are becoming yellow color, and that means the leaves are becoming older. Senescence can be postponed for many days in kinetin treatment in leaves also. That means it can also delay the aging of the plants, aging of the leaves. So senescence is the termination of metabolic activities of plant organ or entire plant prior to death. You see such a beautiful transformation will be seen here. The termination of metabolic activities of plant organs means we can able to postpone the death, or the postpone the leaves falling also. And here the protein degradation and the chlorophyll disappearance are common in the leaves. That means whenever the leaves are or in becoming older, or whenever the leaves are falling, then what is the main process that are taking place in the leaves is the protein degradation will be taking place. As a result, the chlorophyll content will be increasing, and due to this content of chlorophyll, the leaves will not have the capacity for the protein photosynthesis. As a result, the leaves will be falling. So this is also delayed by this hormone. And synthesis of RNA and protein. Here the protein degradation. So here the stopping of protein degradation means we have the RNA and the protein synthesis must be taking place. So cytokines regulate the synthesis of RNAs and the proteins. As a result, the chlorophyll is not degraded, and the leaves are not going to the older condition. And flowering. Cytokines can be used to induce flowering in short day plants. That means the plants which will receive less daylight in order to get the flowering. So this short day plants, when the less daylight or the below the critical light is received by these plants, we are seeing the flowering. So that flowering is also will be influenced by the cytokines. And very important is Morphogenesis. Morpho means the morphological organs are also helpful in the developing in the embryonic stage. That means cytokines is the ability to cause morphogenetic changes in an undifferentiated callus. That means we already know that by the tissue culture from due to totipotency nature, the cells will having the capacity to give rise to the undifferentiated mass of the cells. That is considered as the callus. From this callus, we can produce plants which is having the root system and the shoot system. So the organ differentiation is also be taking place by this the influence of these hormones. And at last is the abscisic acid. This abscisic acid is a growth inhibiting hormone that affects the plant growth. This is considered as the growth inhibiting hormone also. Even though it is having the negative characters, but it also play a very important role in the plant itself. It was discovered by the Kahns and the Endicott in 1963. It is also called as dormine. Dormant means in a resting position or in a sleeping position. That means it is causes the dormancy. Dormancy in the seeds. If you see that, if we also, if we are also not having any exciting way of thinking. Exciting way of thinking means, but you are all the students or you are all the members are going to crack the jobs in the next or the following months. So what we have to get that exciting nature. You must be there. If you're not having any exciting nature, you will be in dormancy condition. You will be in sleeping mode. You won't be any active way of thinking or active way of doing your timetable. So that means we must not be in a dormancy condition. So this abscisic acid is synthesized in the leaves. It is found in abundance in the dominant seeds and the buds. So chemically, abscisic acid is So the physical role of abscisic acid. So first one is the abscission. Abscission means the layer is formed between the leaves or the fruits or the flowers. This layer, what the layer will do means it won't conduct any water or food materials to the organs like the leaves, flowers and fruits. What happens? A dead layer is formed between these leaves and the plant, flower and the plants and the fruits and the plants. This is called as the abscission layer. This is also caused by this ABA, abscisic acid. So it is having some negative role will be seen here. So this abscisic acid is also considered as a growth inhibiting hormone and serins. So ABA cause aging in the leaves and inhibition of the growth. That means it will inhibits the growth of the buds also, and it will be inhibits the falling of the leaves, and it will even inhibits the falling of the flowers. And anti-gibberellin. So ABA inhibits gibberellin stimulate growth. So ABA is also called as anti-gibberellin activities. So this whatever the gibberellin is stimulating, it is showing the negative role. And inhibition of germination. Here the gibberellins are helpful in the germination of the seeds, but it is inhibition the germination. So this due to the inhibition of the germination, we are not having, the seeds will be in dormant condition only, and there won't be any seedling and there won't be any crop yielding for the persons. And the main important point is the stomatal closure. And this abscisic acid is very important, playing an important role in the closure of the stomata. And we see that in some plants, in order to overcome the transpiration, the stomata will be closed here. So, but due to this heavy sunlight or this, the plants will be on shrill, they will be in shrill condition. So in order to close the stomata, these when we spray this abscisic acid, that will be, the stomata will be closed, and we can protect the plant from the hot stress also. And the next one is the ethylene. So ethylene is a ripening hormone, and it is a plant gas hormone. These are also called as the gas hormone. It is produced in stresses by almost all the plant tissues. In nature also, if you see, if a bowl of tomatoes are there, which are in unripe condition, if you keep a one ripe a tomato in that, in 1 or 2 days, remaining tomatoes will also be in ripened condition. But what we are doing in our nowadays, in order to ripen the mango, the carbates we are using, these are the chemicals, they are very harmful for our body. And this hormones is a gas hormone. Denny discovered that ethylene gas was highly effective in inducing fruit ripening of the fruits by producing these hormones. This helpful for the fruits will be in ripen at a same time. So that is the formula for this ethylene, and synthesized in the plant from the amino acid methionine. This is the important point. So it is formed from the precursor is methionine. It is synthesized in the ripening fruits, flowers, leaves, and the roots. And when we see the physiological role of this ethylene, this will be fruit ripening. The important effect of ethylene is bring about the ripening of the fruits. At the same time, we'll see all the fruits will be ripened by this having this ethylene gas. And permeability. Ethylene increase the permeability of the tissues of the fruits. That means the epicarps or the outer layer of the fruit will be becoming very soft. As a result, they increase the permeabilities of the tissues also. As a result, the ripening will be seen here. And flowering. Flowering can be induced by application of the ethylene. So, if the plant is having the vegetative growth, a plants lo mottam kuda vegetative growth mana chusina flowering ane lekapothe, there won't be any use in the commercially. We are not having any use. So, at that time, this ethylene can also induce the flowering also. As the flowering, the fruits will be formed there, or there may be a vegetable. If you think that if the plant of a vegetable plant is there, the flowering, we are not having any flowering, just the plant is growing, but it is not giving flowers or any vegetables or any fruits. We will see, and after some days we will be pluck that plant. So that so nature itself is having that ethylene hormone in order to bring the flowering in the plant itself. And abscission, it stimulates the formation of abscission layer in the leaves, flowers, and the fruits. And in some leaves, if the leaves are older in condition, they are not having any photosynthesizing metabolic activities. So that leaves the wall formation, the abscission layer is forming, and the leaves are falling itself. So these sometimes they may be useful, or sometimes they may be affecting the plants also. And apical dominance. The mainly apical dominance is seen by the auxins, and ethylene with auxins causes apical dominance in plants also. And climatic respiration. During ripening, there is a sudden rise in respiration in the fruits. It is called climatic respiration. It is brought about by the ethylene. This type of respiration is seen by the ethylene itself. That means if all the fruits are keeping in one bowl, due to this respiration process, all will be, the gas will be spreading, and these are helpful for the total climatic respiration that leads to the ripening of the fruits also. And softening of fruits, due to the ripening condition, the epicarp will be soft. As a result, the coloring of the fruit will be changes. and rooting. Ethylene induces the rooting of the cuttings also. If we are having the root cuts, for example, if you take some roses plants or any root cutting, if we implement this ethylene, they are having the formation of the roots will be seen. That means due to the formation of the roots, the plants will be accustomed to grow in that soil easily and withstand and synthesizes of enzymes. So ethylene stimulates the synthesis of enzymes like peroxidase and polyphenol oxidase. This will be helpful for the fruits to be ripened. And the senescence. This also is helpful. That aging of the plants will also be stopped by this cytokines. Okay, students, thank you.
Okay, regarding of this class, have you any doubts, my dear friends? So, madam is ready to clarify your doubts. Any doubts?
Peroxidase and polyphenol oxidase.
Polyphenol oxidase, madam.
These are the enzymes. These are the enzymes are initiated by the cytokines. They are helpful for these enzymes helpful for the ripening of the fruits. They are produced in the plant itself.
Okay.
These hormones will initiate these enzymes to be released in the plants.
Okay.
Anybody
Actually,
else?
they are produced from cytokines, right now? Is it now?
Okay, anybody else? Okay. So, madam, really, today's class is a very excellent class. You have presented wonderful and a very clear-cut idea about the plant growth regulators from auxin to ethylene, auxin, cytokines, gibberellins, abscisic acid, and ethylene. All of these are all the plant hormones which are induces the growth and control the growth and development of the plants. So plant physiology, this is the one of the important area for the scoring of the all competitive exams, my dear friends. I hope you clearly understood today's class, and also we will send this link in the YouTube link, also send in WhatsApp group, my dear friends. Really, thank you very much, Madam Padmaja, Madam Gar.
Thank you, sir. Thank you, sir.
Really a wonderful presentation.
Thank
And
you,
you spare your valuable time in this hot summer and midday. So this hot time, you are clearly explain and spare your valuable time for our young aspirants, those who are going to attend CPGET 2023, and also some of the lecturers who are working in various colleges, from junior colleges and degree colleges and Gurukula degree and junior colleges. Some of the guest faculty also joined, and they are enlightened from your class today, madam. Really, they are very happy. They felt very happy because of your explanation is wonderful and very clear-cut idea about the plant hormones. Okay, madam. Thank you. So tomorrow also we have class in the same time. Okay. Is it okay, madam?
Okay, sir. Okay.
Right, madam. Thank you.
Thank you, sir.